This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 98, recorded on October 29th, 2015. Hi, everybody. I'm Vincent Racaniello, and with me is Dickson Despommier.
Hello, Vincent.
And Daniel Griffin.
Hello, Vincent Dickson, and everybody who's listening.
And
hello to me, too.
I meant to say that, too. But
Yes.
You want to say hello to yourself?
That's the way I don't hear.
Why not?
Nobody else will, right?
Well,
Yeah,
It's a lovely
nobody else
day.
will.
It's
a
a lovely
gorgeous
full
day
day.
out there.
You know, we have gone through cold weather, and now it's warm again.
That's right. But it won't last.
It's sunny and 20 degrees Celsius.
This is the blast from Patricia.
Really?
Yeah.
What is Patricia?
Patricia was the largest hurricane in the northern hemisphere, I believe, ever recorded. Although I thought the one that struck the Philippines 2 years ago was the largest, but it might have been then. But I think this one was even
Dickson,
bigger.
when there's a hurricane in the Pacific, what's it called?
Cyclone.
And what's a typhoon?
A cyclone.
Same thing?
A cyclone is a hurricane, is a typhoon. They're all the same, basically.
It depends where they occur, right?
That's true. That is true. This one was classified as a hurricane.
Which do you like better, hurricane, typhoon, or cyclone?
I don't think I like any of them,
All right.
actually.
Daniel, you've been seeing patients, right?
I have
Does
been.
this put you in a different mindset?
Yes. Well, I should say I've been seeing more patients and doing more clinical the last week or two here than, usually I don't do quite as much. But no, every morning I've been getting up at 5:30 in the morning, and I've been heading over there to what do we call our hospital here? Is it Columbia Hospital? Is it Presbyterian?
Well, the medical center.
CPMC.
The medical center is Columbia University Medical Center, and the hospital is Presbyterian Hospital still.
Okay.
So Columbia Presbyterian Medical Center.
No, you are so out of it, dude.
That's how it used to be referred to.
Yeah, but that's how it used to be, Dickson.
And when did that change?
Because things used to be a certain way.
200 years ago?
No, the medical center paid a company to rebrand it.
Oh, I see. And
I
when
don't know
did
where
that
you
happen?
were. It was about 8 years ago.
8?
So it used to be Columbia Presbyterian Medical Center, CPMC. It used to be on my ID. Notice my ID now says Columbia University Medical Center. The name of the hospital is still Presbyterian Hospital. They didn't rebrand the hospital. All right? Now, what they should do is get someone to give $500 million to rename it, like Carl Icahn Medical Center. Would you work for the Icahn Medical Center?
Only if he's iconic.
Like the Bloomberg University, what was it? Johns Hopkins Bloomberg Medical Center, something like that.
No, it's School of Public Health.
Public Health. The Weill Cornell
We have
Medical
the Mailman
Center.
School of Public Health.
So that Weill Cornell is here in the city.
That's true.
But here they
We
can't
have
get people to
Milstein
give money.
Hospital.
They told me once the reason people will not give money is because you're here.
It's all Dixon's fault.
That's probably true.
But for example,
so call it CPMC.
It could be the Griffin Columbia University Medical Center.
Could be.
For $500 million, you could have that.
I have to check my bank account. I may not have that much. No, I should say, so I do a lot of outpatient, but then periodically I do these stints of inpatient work.
What does inpatient mean?
This is where I've been on TWiP too long and I just can't handle it now, and I become inpatient. No.
Instead of being able to go home at the end of the day, you have to stay there.
No, it's a focus on the care of individuals who are ill enough that they require hospitalization.
Right.
And an outpatient comes for a few hours.
They come to a clinic. They're seen
more like 8 hours.
Yeah, 8 hours at
Before they
Columbia.
get seen. That's right.
A little, maybe 5 minutes with a provider.
So you're doing more inpatient now.
More inpatient, and it's Columbia has a great traditional academic model where they actually have two attending level. So you finished your training, board certified. So I'm working with another one of the infectious disease doctors, this Ellen Morrison. It's kind of your classic infectious disease doc, sort of that combination of intellectual, very knowledgeable, but sort of that compassionate aspect that I like to think is common to the ID community. And then they have 2 residents who are kind of more experienced post-MD trainees, Shreya and Brian. And then we have 3 interns. I wrote the names down because I can't remember all this, but Christine, Andre, and Nick. And they're, they've just finished. They're out of medical school. Here they are for the first time, really making these decisions. And then we have a couple medical students. This Daniel, who's here from Nashville, Tennessee, and Joshua, who's actually a Columbia local medical student. So you have this whole group, and you go around, and traditional. You see the patients, you do the bedside exams, you discuss, what are the teaching points. It's a great experience.
And these are AIDS patients you're seeing.
I will say about half of them are infected with HIV, and then they've thrown in. I always say I signed up to take care of the HIV patients, but every so often we take care of patients who have infections, other infectious agents.
What is the most common other infectious agent that you see?
It's such a mix.
Not C. diff.
No, we try to stay far away from C. diff. We had a big discussion this morning: is how can we minimize our patients getting C. diff? No, but that is a huge problem. Usually something that they don't get us called for.
Yeah. How about staph infections? Are they common?
Staph infections are super common. One gentleman who had a gentleman with kidney failure. He's on a dialysis, and he has what we call a fistula. It actually looks like he has a staph infection of his fistula. Some great infections.
And strep pneumonia, I guess.
Still super common.
Yeah.
Seen any whooping coughs?
No.
Whooping coughs?
Because there are a lot of people out there that are not vaccinating their kids.
Yeah, no, that was quite an uptick for a while. I always tell people, do you want the vaccination to prevent the 100-day cough? And usually people say, yes, I do not want to cough for 100 days.
How about Klebsiella?
Klebsiella, particularly a problem because of the drug resistance.
Right.
And we see that. Pseudomonas, definitely.
Have you run out of the bacteria you know, Dickson?
No.
Name another one.
One more, Vibrio.
I knew he was going to do cholera. I was going to say that.
You get cholera here in New York City, Dickson? Imported cases?
They arrive here with it. They didn't catch it here.
What about this new one that's popping up? Has a really interesting name, which is escaping me.
MRS?
No.
Flesh eating.
Oh boy. I talk about this on TWiP a little bit. It's more prevalent now than it ever was, and it's an unusual Acinatobacter.
Yes, Acinatobacter. Again, another organism that can have a lot of drug resistance challenges.
Right.
Any fungi that you want to talk about?
We definitely see a number of fungi. Aspergillosis, we were just doing a little teaching with them on that.
Candida.
Definitely
in
Candida.
Patients,
we just had some Candida growing in somebody's blood.
Coccidiomycosis? Not here, right?
No, Southwest.
More a problem in the Southwest.
But you, even in Colorado, you didn't see that.
More Arizona and New Mexico. Yeah. So what we have is we used to call them the... what do we call these? The people that would go down there, the winter birds or something? Snowbirds.
It's called valley fever, right?
Yes, the acute.
What about histoplasmosis?
Histo, right.
Histo, interesting enough, everyone's always taught that this is this Ohio River Valley, but it's really prevalent throughout the Caribbean and certain areas of South America. So a lot of the histo cases that I have seen have actually not been native U.S., but people who've come from the high-incidence Caribbean areas.
How about Naegleria fowleri?
Now we're getting personal.
Well, maybe we should go to our case at our emails. Is it about time?
It's a little ID lesson there. Okay, very good.
Here we go.
We have no follow-up, so we can go right into our case.
Okay, that's great. Well, let's remind everybody, or for those people who are tuning in for the first time, we're about to solve 2 cases. We had 2 patients, a 12-year-old girl who presented for care with a painless dark brown area on the right knee, giving us the story of brothers and sisters often playing together, frolicking about. Hadn't reported any specific major trauma to this area that had been expanding, and then later on goes to develop these cognitive changes, and we have an abnormal MRI. There's also a 5-year-old boy from the coastal region of Peru. Both these cases are from Peru. And he's brought in by his mother with a red swollen area on his face. The mom gives us a story, as we said, of a scratch on the nose followed by swelling, which extends to the right cheek. with some nasal obstruction, wasn't responding to antibiotics. And he has also got some sort of infiltration on his brain scan. And we had a lot of people with I would call them educated.
They're catching on to
entries.
So the first one in was from Robin, who wrote Balamuthia mandrillaris and sent some links to documents, one of which asks, is Balamuthia a public health concern worldwide? And the next one was from Jan, who writes, hello, gentlemen. Sorry to have been remiss to answer a few case studies, not through lack of appreciation, but I've been too busy. After consulting Dr. Google and my second hand copy of parasitic diseases, I think it's chronic Chagas disease. The patients may have contracted it by scrapes and scratches, which got contaminated by fecal droppings whilst playing, not through the kissing bugs. Greetings and thanks again for the podcast. Jan from Grey Overcast, Rotterdam, 14 degrees C. P.S. Only 4 copies sold of People, Parasites, and Plowshares. Does that make mine a collector's item?
Oh, we've sold more than that.
You know, Dickson, last time you joked about selling only 4 copies, and you should really be careful because people take you seriously.
After listening to all of this, they've taken me seriously? I can't believe that.
How many copies have you sold? 10 billion?
I have no direct numbers on that, but they printed 3,000, and they're getting near the end of that run.
Okay.
So we might have sold 3,000.
Okay.
Not me. That is Columbia University Press might have sold with.
Are they going to do another
3,000.
printing?
No idea.
All right.
But yes, no matter whether it's 4 or 3,000, you have a collector's item.
Take the next one. From Frederick.
Okay, Frederick writes, greetings. My guess would be amoebic encephalitis with an acanthamoeba species. I can't say I have any clinical experience of this, but should it be correct, the prognosis seems rather grim. Frederick.
Daniel.
Dr. Wink writes, Daniel, I appreciated the very interesting cases and most difficult yet. I think these 2 Peruvian youths were infected with Balamuthia mandrillaris. This is a free-living amoeba that can be isolated from soil. A skin lesion may be pigmented and precede other symptoms by weeks or even months. A granulomatous encephalitis may develop that can appear infiltrative on MRI. I certainly didn't know about this until I investigated. Thanks for the challenge. Wink Weinberg, Atlanta.
Nice.
Andrew writes, greetings, honored TWiP doctors. I have brought Dr. Google in for a consult, and we suggest that the children have been infected with Balamuthia mandrillaris, a free-living amoeba. The typical skin lesion is painless and dark red or violet. I refuse to use the word violaceous or violaceous. The amoeba may migrate to the brain, causing a condition known as granulomatous amoebic encephalitis, which may involve paralysis and seizures. GAE is usually fatal. There are no treatment guarantees with B. mandrillaris, but there has been some success in the past with multi-drug combinations of antibacterial and antifungal medications. I hope these kids were okay after treatment or that I am wrong. Yours, etc. Dickson.
Zachary writes, greetings, Daniel, Dickson, and Vincent. I was initially thinking primary amoebic meningoencephalitis, PAM, caused by Naegleria fowleri for the case of the week. This organism is well known as the brain-eating amoeba. The route of infection for Naegleria is through the nasal cavity. I remember living in the South several years ago and reading a story of several unfortunate individuals from Louisiana, which happens to be my home state also, who used tap water in their neti pots and unknowingly infected themselves with this brain eater. Needless to say, people with PAM do not have a high survival rate, about 95% fatal, he lists. The smoldering aspect of the infection in these 2 cases leads me to believe that they have contracted granulomatous amoebic encephalitis, or GAM, the causative agent being either Acanthamoeba histolytica, or Balamuthia mandrillaris, with B. mandrillaris being the more likely culprit due to the length of time since infection. I hope I am correct, but cannot be sure. I have discovered that almost all parasitic infections can somehow end up in the brain, albeit rare in some cases. I think of nothing more horrifying than a parasite infecting your brain, and I'm suddenly stricken with the image of Ricardo Montalban ordering a parasitic earwig to be placed into the ear of Ensign Chekhov for the penultimate Star Trek movie Wrath of Khan. It is a pleasant, sunny and 18 degrees here in eastern Washington, but I fear that winter is not far. Keep up the great work on TWiP, Zachary.
Stewart writes, Dear TWiP-lets?
TWiP-lets.
Yeah, we are.
That's good. TWiP-lets. I like it.
That was clever. My name is Stewart. I'm a sexual health doctor on the Gold Coast, Australia, but I have loved parasitology ever since I was a medical student. I recently had the privilege of returning to university to study infectious disease epidemiology as a postgraduate student. Of course, I took medical parasitology as one of my coursework electives and rekindled my passion. My 5-year-old son has become fascinated with parasites too. After getting hold of my textbook and from overhearing snippets of your podcast. He recently dressed up in my clothes, strapped a bag full of books to his back, and headed out the door, pausing briefly to inform us, I'm going to university to study tapeworms. That's my boy. My diagnosis for the case is granulomatous amoebic encephalitis caused by Balamuthia mandrillaris. With luck, you'll see trophozoites in the biopsy, but if not, granulomas and multinucleated giant cells would be supportive of the diagnosis. DIF or 16S ribosomal RNA nucleic acid amplification testing on the biopsy tissue should confirm the presence of the organism, or alert you to the need to search for an alternative free-living amoeba like Acanthamoeba or Sapinia. The outlook for these two kids would have to be poor, particularly for the 12-year-old girl. She already had signs of raised intracranial pressure. Hopefully the little boy's prognosis will be better, but would still be far from hopeful. It's sobering to remember that as my little boy fills my heart with joy when he pretends to go off to study tapeworms, there's another little boy and another dad on the other side of the ocean who face a very different future. My next stop is the Gorgas Institute's website, where I will take up Daniel's excellent suggestion of donating. Thank you for all the TWiPs. They are an excellent resource, Stewart. Dr. Stewart Aiken, Senior Staff Specialist, Sexual Health Medicine, Gold Coast Hospital and Health Service.
Wow.
The Gold Coast of Australia. There's Gold Coast University.
There is.
Yeah, I interviewed some people from there.
It's an interesting place. It's likened in demeanor to Florida.
What did you say? They have lichen there?
No. It's made. There's an. Let's try this again.
I heard what you said.
I think you did, actually. You're just putting me on.
Yes, I am. Paulette writes, Hello, Doctors Racaniello, Despommier, and Griffin. Thank you for the informative and entertaining podcast. You clearly put a lot of time, effort, and thought into your work, and we listeners appreciate you sharing it. Dr. Griffin's Peruvian cases that presented skin lesions followed by an infiltration process in the brain are a match with the description of patients suffering from an infection by Balamuthia mandrillaris as described in an abstract. I couldn't afford to read the article
behind a page
in the American Society of Tropical Medicine and Hygiene. The article, A Novel Therapeutic Option for Balamuthia mandrillaris Infection by the authors, describes an uncommon disease involvement of the skin with subsequent extension to the CNS, and 2 patients whose disease began with skin lesions on their knees and one with a lesion on his nose. Wow. It's a big world, and it may be just a coincidence that this article describes many of the same symptoms and details as Dr. Griffin's cases. I hope that it is coincidental, because this sounds like a truly awful disease with a grim prognosis, and Dr. Griffin's cases concern children. But it does seem to be a match. Yours, Bob G. Dickson.
Here we go. Venkat writes, Hello to the TWiP team from South India, where the nasty dengue wave is starting to rise high again. I recently started listening to your podcast, and my favorite is TWiP. Parasites always are so fascinating, mainly due to their interesting presentations. After several weeks of listening to TWiP and solving cases, I was confident that I will nail the next one. But TWiP 97 was tough. Both presentations are not classical of any particular disease, at least to my knowledge. The MRI seems to suggest tinea infection, particularly neurocysticercosis, but I am not fully sure, as the other pieces of the puzzle do not fit. Also, the long timeline since onset to clinical visit and the painless brown lesion on the girl's knee makes it even more complicated for me. I am currently undergoing training in infectious diseases, and your podcasts and lively discussions are such inspiration. Keep up the great work. Venkat B.
Annie writes, Dear Sirs, I just read about this amoeba a few weeks ago. I vote for Balamuthia mandrillaris. Hope I spelled that correctly. Still your biggest fan, Annie.
Nice.
And the last one is from Jeff, who writes, Dear doctors, I listened with interest and shortness of breath podcast while exercising to your case from TWiP 97. These two cases corresponded to a case I saw while at Universidad Peruana Cayetano Heredia in Lima, Peru in 1997. At that time, it was considered quite a zebra, and no one knew what to think. 2 years later, I saw two more cases. At that time, however, Dr. Eduardo Gotuzzo had a small case series, and it was known to be caused by Balamuthia mandrillaris. At that time, it was intellectually quite satisfying, but tragic for the patients, for no treatments had been found to slow the slowly progressive infection, and all these patients died of the infection. I understand that miltefosine, a drug used initially for cancer, which was repurposed for leishmania, has been found to be effective. I am sure you will fill us in on treatment. I am attending the American Society of Tropical Medicine and Hygiene meeting in Philadelphia, where it is gray and 20C, a perfect setting to hear about the explosive growth in knowledge in the field of tropical medicine. Please keep up the wonderful work. Jeff is Jeffrey Jones, MD, MPH of Indianapolis, Indiana.
Nice. Do you know what he meant by zebras?
Yeah, I do.
Would you like to explain that?
When you hear hoofs, think zebras. Think of the unusual.
No, it's the other way.
It's think horses, not zebras?
That's exactly what
So don't think of the unusual, think of the common?
Yeah. So
when I see you, I think zebras.
Well, you should.
So how does the full saying go? How does the full saying go?
The statement is, when you hear hoofbeats, don't think of zebras, think of horses. But that's true where horses are common and zebras are not. In Africa, it would be just the opposite, of course. When you hear hoofbeats, you might think of gnus or, eland or, of course, the wildebeest as well. So horses are not there, but you would certainly think of zebras there. But in other words, consider the obvious first. If that doesn't pan out, then go to something unusual. But don't go to the unusual first.
Okay. Got it.
But this is a definite zebra for those people, if you haven't never heard of it before.
Well, what would be the common thing?
Oh, well.
In New York City, if you hear hoofbeats?
No, but
I think we'd be at a loss.
Think of a policeman or a carriage that goes through Central Park, but don't. No, I think if you're living in Indiana, the common one would be Naglaria. That's the way that would work out.
Got it. All right. So, Daniel, here we are. We had a couple of Balamuthias and a few other things scattered in there.
Right.
So what else did we do with these two kids? You were there. You took care of them.
Yeah, well, I think, as I mentioned, we did a biopsy. First we went through what we call our differential diagnosis. I think it's been a while since we used that terminology, where we go through what could it possibly be and then try to narrow it. And they listening to an audiobook recently, actually, about a Columbia medical student's first year. Actually, he went to Harvard Medical School, then came to Columbia to do his internship and training. He makes the quote that while you're in medical school, you learn how to generate a differential diagnosis. And then once you finish medical school, you learn how to narrow it. And you still go through that process. So we started off with the fact that, okay, we're in Peru. What makes our hoofbeat noises? What looks like this in Peru? It was really long. It was slow. So we started to think of fungi. Which sort of fungi might be involved here? So paracoxy is a particular fungi that's down there. So we thought about that. But then where were they in Peru? You see more paracoxy if they're, sort of the eastern side of the Andes. These people were, as I mentioned, coastal, not from that area.
Right.
The boy had this lesion on the face. Rhinoscleroma is something that we sometimes see down there.
I'll be darned.
But he's too young for that. Cellulitis, but again, too slow, too indolent. Yeah. But then we end up with the issue that he's got, and she has, a skin lesion, very, very slowly progressing, and the brain infiltration. And as noticed now, years and years ago when this first started happening, nobody really knew what it was. Yep. Now there's been an experience, so the biopsy was done, and the pathologist is going to tell us what the result is. And actually, I'm going to say, in this case, the sample was even sent to the CDC to help us pin it down.
Oh, nice.
So I don't know if Vincent or Dickson, you had any more questions, or if you want me to tell you the pathology.
Well,
What do you think it was, Dickson?
Well, my initial thought was Acanthamoeba, but it's obviously not, because Acanthamoeba doesn't present as a skin lesion. It's just not going to happen like that. And so it was a scenario that I had not incorporated into my own teaching. So therefore, I was less familiar with it than I should have been, because I should be keeping up no matter what. That's a self-admission. But I think the correct diagnosis would be Balamuthia mandrillaris. And by the way, I must mention that the word Balamuthia is an honorific term for a gentleman by the name of Balamuth, who was a world expert on pathogenic amoeba. So they've actually named this organism in honor of his hard work back in the 40s and 50s. And as a technician, we actually used his book as a source for differentially diagnosing things that we saw under the microscope. So I guess if he were still alive, he would be honored to know that they've named an organism after him. But so my guess is Balamuthia.
Okay, so you said you did a biopsy of what?
Of the skin lesions in both cases, not the brain. So
yes,
in both cases, there was a biopsy.
And then did you look at this initially?
And you could see, I actually have the picture.
And then after you looked at it, you sent it to CDC?
So sent to the CDC for typing to tell us a little more about. So we knew roughly what kind of, and I should say, it was an amoeboid form that was seen
in both cases.
In both cases, and then it was sent to the CDC for typing. What sort of amoeba was this?
Okay.
And yes, I'm always impressed by our listeners. They got it.
Balamuthia. Google was a big help.
So I had thought last week it was, or when we recorded last time, it was an amoeba of some kind, but I didn't know what kind it was.
Right.
And now there's been a number of cases of this described down there in Peru. So there's different amoeba in different environments, and then the different amoeba can present a little bit differently. Now, the Naegleria fowleri, which is what I think U.S. physicians are familiar with, there's been a number of cases in the South, but I'll go even farther up. Virginia, I know a doc in Virginia who's seen some cases. There's certain areas where certain situations that result in people getting this infection. And the infection actually tends to invade through the cribriform plate. Right. Mm-hmm. So it's going to go up your nose. It's going to go directly into the brain, which is different than in this case. We have this long-term infection in the skin, which then ends up spreading to the brain.
So another thought I had along those lines was dermal entamoeba histolytica. Now, I know that sounds kind of like a far reach for this diagnosis, but I remember hearing Dr. Michael Katz, who used to be the course director for parasitic diseases here at Columbia, give a lecture once to physicians about his own experiences in travel. And one of them was in India, and he encountered a woman who was walking only on the balls of her feet. She would not put her heels down.
She thought she had high heels on.
Well, she couldn't even put them on, because if you looked on the bottoms of her feet, her entire heel was eroded away. It was an open ulcer. And this open ulcer turned out to be caused by entamoeba histolytica, because when they biopsied it and they typed it out, they identified this as the organism. Wow. And she caught it by the habit of course, everybody has to defecate, right? So she would go into an alley of some sort and defecate, and she would, of course, be barefoot. And she obviously had encountered some rough surfaces with her foot and had an open lesion due to the just a cut of some sort. And this gave entry to the amoeba, which she also had in her gut tract.
Correct.
So that's a little bit different than what we see here. But then I thought that was too unusual for two people in the same place in Peru.
This is a soil dwelling.
Yeah, that's right.
It's globally distributed.
That's right.
Do you know why it's called mandrillaris, Dickson?
Well, what is a mandrill?
It's a kind of monkey, right?
It is.
And that's where it was isolated from.
How about that?
By your friend Balamuthia.
Yep.
Whatever his name is.
Mandrills are native only to Africa, as I recall.
it is globally distributed, but there have been only 200 cases described worldwide, 70 in the U.S.
Right.
How about that?
Well, that's probably more than Naegleria. But I'm fascinated by the growing list of normal organisms in the environment that have sought out refuge in the human host as an accidental parasite, and this is another one of those examples.
I'm looking at a CDC site, and it says the CDC is available to help diagnosis, and that's just what you guys did.
Yeah.
Now,
and it was a ribosomal.
it was a PCR.
Yeah, that was done. So it's PCR, the NAT, the nucleic acid amplification test.
By the
Interesting.
By the biopsy, you could not type the amoeba. Right?
True.
It was just an amoeba, right?
Right.
Is there any evidence or suggestion that these kids were immunocompromised at all?
No, there wasn't.
So these infections are occurring in normal individuals?
In people who we think of as immunocompetent. You know, we talk a lot now about how little we know, and how when people start getting full genome and exome sequencing, I think we're gonna start to realize that when you've got 100 people and 2 people get sick, there's probably some reason why those 2, or in this case, when you have millions of people and 200 get sick, what was it special about them?
Sure.
And I'm optimistic that we're actually going to start figuring this out. And so when somebody comes in, you'll sort of know, hopefully ahead of time, what to worry about, and maybe ahead of time what to worry about so that they don't get these things.
Right.
But in this case, I think the idea is somehow you have a break in your skin, you have a break in that barrier that protects us, and the amoeba is getting into our tissue. I don't know how often that happens, that a normal person without a sort of chink in their immune system armor would not be able to control this, but I'm not sure. With Naegleria, we're thinking the other amoeba that we see in the U.S., we're thinking that physically gets in.
Yeah. I appreciate Vince's question, because every little kid plays in the dirt.
Yeah, exactly.
Every little kid gets scrapes and scratches all over the place. Exactly. Workers that are doing road work or construction work, they always come home injured. How come they're not getting sick with this
good
thing?
question.
Or are they misdiagnosed? Maybe they're misdiagnosed because it's such an acute infection.
Yeah, no, I'm on board with the idea. I think that this is probably getting in. So this, people are always falling down and getting scratches. There's some reason why these people could not fight it off.
So how long do you think they had the skin lesions before it went to their brain?
Well, in these cases, it was many, many months. And if you go through, now that we have an experience with this organism, it's a long period of time.
So a question about the lesion in the skin, then. What does that look like? You didn't do a very good job.
I did not. I did not. I should have brought my notes from when I described it.
Yeah, well, give us a more complete description.
I thought for the woman, I described it as a dark...
yeah, but was it craterform? Was it oozing? Was it open?
No, it was not open.
So why did you say it was a lesion then?
Let me take a look here.
I always think lesions as being open sores.
French foreign lesion.
French foreign lesion. Is that?
That's right.
Let's see how I described it.
Because if it was just a darkened area on the skin,
it was a painless dark brown area on the right knee.
You see, who's going to report that to anybody? I mean, little kids won't come home and say, "Mommy, I've got a brown little thing on my knee."
Well, it was quite impressive. And the reason so initially it was pretty minor, but it kept expanding, and there was swelling. So there was this enlarging, dark, swollen area around the right knee that's developing over months.
If a dermatologist had seen that, what would they have thought?
I guess it depends how good they are.
No, is there any other?
Hopefully they would have said, I don't know what that is, and I'm going to get a biopsy so that I can figure it out before it goes to her brain. Yeah. But in all reality...
I mean, it didn't look like a melanoma.
You know what they would have done? They would have rubbed some cream on it and hoped it got better, and then tried cream 2 and cream 3. And when cream 3 didn't work, maybe they would biopsy then.
So, I mean, it doesn't look anything like melanoma, for instance, or for some other skin dyscrasias that...
Melanomas are little guys.
No, but they grow. They do grow.
Well, if they grow, by the time they're this big, you're
probably going to be dead. But this was growing too. So I just... That's very puzzling as to how this presents, that's all. Because apparently the wound then heals, creates an anaerobic environment. A lot of these amoeba are anaerobes, by the way, because they have no mitochondria. And so the next thing you know, you've got this slowly growing lesion because it takes a long time for it to start. right? It's almost like a shagoma.
I'm going to show Dickson a picture of it.
Oh my God! It looks like you got into a fight.
Yeah, it's pretty dark. Would you say purple?
Purplish, brownish, blackish.
Would it be
No, tell me that doesn't look like a melanoma though. In some parts of it, it looks.
I'm going to show Vincent.
Violaceous?
No.
It's really more blackish, right?
It's very black.
It looks like a hemorrhage of some sort.
Gangrene.
No, it doesn't.
New York Jets
It doesn't really look
defensive team.
necrotic,
That's
but
what
it is dark. I'll say.
Very dark.
Yeah,
Yeah,
so
that mother would respond.
At what point would the mother say, I think there's something going on here that I don't
Well,
I don't
again,
understand.
depends how good she is, but
Well,
if it
those kids
persisted-
are not complaining.
if a bruise persisted for over a week, I would think that most mothers would say, "Let's go get this checked out."
See, if your dad was a doc, he'd say, "You're fine." And you just
My father did that all the time, and that's why he died, because he did it with himself.
Unfortunately.
Yes.
Unfortunately.
Instead of
Well,
fun.
let me give you guys good news, though. Now, one of the advantages of there being a number of cases of something is that we can actually start trying things and seeing if something works. And as some of our emailers intimated, the early experience with a lot of these amoebic infections was really dismal, 95% mortality. And it's really great, actually, that people mention miltefosine, which we've discussed a little bit. So these two individuals were treated with this combination of albendazole, miltefosine, and fluconazole.
Look at that.
And that actually is something I think, I don't know if that's in that Eduardo Gutazzo.
That's an antifungal, fluconazole.
Fluconazole is antifungal. It's an azole, but it also
kills off leishmaniasis, dermal leishmaniasis.
That's right. It is.
Yeah.
I remember that from the parasite.
So we'll say it's antiparasitic as well, right?
That's right.
We could say that.
It might also be well, okay, go ahead.
No, you go.
No, I was just going to, there are some drugs like metronidazole that attacks all anaerobes. It doesn't matter whether you're an amoeba or a bacteria, it'll kill you. So maybe this drug has a similar mode of action with regards to a class of metabolic organisms.
They were treated with oral medication, is that correct?
They were treated with oral medications.
For how long?
And they both got better.
They were both started initially on an amphotericin and then switched to oral medicine. And when I saw them, they are doing well. Currently doing well.
Right.
We'll have to see over time. I don't think we know enough to know whether or not you can ever truly eradicate this from the body, or if you're just going to basically get it into a suppressed, controlled. And again, as people are commenting, we're getting experience with these infections.
I mean, this is a new thing. I didn't stop teaching that long ago. I did go to all the meetings.
In terms of progress, it was ages, Dickson.
No, 2009. That's not that long ago.
Yeah.
That's only 6 years.
You haven't read a paper since then.
That's not true. No, a paper. I've read many papers since.
I have to be nicer to you. I'm sorry.
Yeah, you should really be much nicer to me, Vincent. Because I scan the literature regularly, and it's really a new entity.
The trout literature? You scan the trout literature.
Oh, I live the trout literature. So, were you there long enough to see the lesion get better in the skin, at least?
The sort of timing I was able to see before the treatment started, and then we'll see. Actually, I saw the woman a couple times while I was there. But yeah.
And there was an improvement in the lesion itself, or you didn't?
I think it's probably going to be quite a while, to see this sort of changes, because this is a pretty chronic condition.
Right.
Did you do a follow-up scan after treatment to see what
and
was
it was
going
actually
on?
improvement.
It was improved.
So,
all right, good.
But yes, I think it's it's nice to see that we're recognizing new diagnoses in the neglected tropical medicine world and getting improved therapies, and people are actively working on these things.
You bet.
All right.
Yeah, definitely.
All right. The next phase of TWiP.
The next phase of TWiP.
With a paper that was selected by Daniel in a journal called Parasites and Vectors. You know this journal, Dickson?
No, isn't that anything like, I guess, pit bulls and parolees? That's a reality show.
This is an open access journal on Biomed Central, so you can find it if you'd like to take a look at it. It's called RNA-seq Analyses of Changes in the Anopheles gambiae Transcriptome Associated with Resistance to Pyrethroids in Kenya, Identification of Candidate Resistance Genes and Candidate Resistance SNPs. Daniel, why did this paper interest you?
I noticed in our last discussion, we brought up insecticides. We were trying to figure out whether I was saying something wrong or pronouncing something wrong. And the following, was it Saturday or Sunday? I forget which day it was. But I was sailing with a friend, and we were listening. I not only record these podcasts, but I listen, hopefully because I catch something and kind of improve my performance, shall we call it. And I noticed, I said, we probably should talk a little bit about this important issue, because if we can control the vectors and understand how we control the vectors, we can actually have an impact on how many people get ill. And I thought, why don't I look for something up to date, molecular, that's addressing this issue.
Right, and there's a lot of spin-off from this paper as well.
Spin-off?
Yeah, we'll have some other things to talk about.
Really?
We will.
Are we going to cast a wider net on this topic?
Oh, good. Excellent lead-in.
This paper's about pyrethroids.
Yeah.
Did we mention that in the last podcast?
We mentioned the word, but we didn't agree on what it meant.
What does it mean, Dickson? Have you since looked it up?
Pyrethrum is the parent compound, and it's isolated from chrysanthemums.
Correct.
And it's a naturally occurring insecticide that plants make for their own defense. And they're very short-lived, and they're very non-toxic to people, which has two advantages. One is that you can use it as a deterrent for vectors, whether it's a control mechanism or a preventative. That's another issue, and we're going to get to that. And then the other thing is that since it's short-lived, it decays and loses activity, but it also loses toxicity for even high doses of insecticide. Mm-hmm. And so in this case, we're going to call it a double-edged sword, because I think Daniel's reference, casting a wider net, has a definite tie-in to what we want to discuss. So why are we discussing resistance to pyrethroids, which are chemically modified pyrethrums?
So the original was isolated from chrysanthemum, and then they've chemically synthesized derivatives.
Just like penicillin.
So these have short half-lives, so they don't remain in the food chain, as does DDT, for example.
Exactly right. So it's a perfect one that you want to spray on things, or
in fact,
you want to
dip
something.
You can go to your garden store and buy.
You can
insecticides with these in it. Right?
Pyrethroids. That's correct. That is correct.
Now, what is the target of a pyrethroid, Dickson?
It's the insect, Vincent.
Well, I know it's the insect. They're insecticides.
The nervous system.
Usually. The nervous system, and what is the molecular target? Daniel. You're squirming your way out of this.
That's exactly what it is. It's the paravoltage gated sodium channel.
It's a sodium which is involved in nerve transmission, right? Yeah. You read this paper?
I did, twice.
See, so he has read papers.
Only twice.
Exactly. If you count one paper twice, I've read papers.
Yeah. Okay. He's read two papers, this one twice.
And it's my understanding that previously resistance to this, which does arise in insects, has been associated with amino acid changes in this channel, right?
Why don't you go back a little ways, though?
Go ahead.
If you don't allow me to say this.
You can.
And with the original isolation of pyrethrum,
yes, sir,
and the follow-up chemical synthesis of pyrethroids, it became obvious that there was a particular intervention strategy for controlling vectors that related to the use of these agents on certain accoutrements. I'll be vague at the beginning, but I want to come back to it, that you would use at a particular time in your daily cycle to prevent exposure to these vectors, which are mostly night-feeding vectors, by the way.
You're talking about Bednets?
Yeah, I'm talking about Bednets.
Why don't you just say Bednet instead of all this beating around the bush?
Because there's a huge program, a huge international program, and one of the proponents of that program is Jeffrey Sachs on our downtown campus. Yeah. And he spent most of the last 8 years or so trying to raise money to fund a Bednet program for most of sub-Saharan Africa and India. And he was successful, by the way. And it turns out that the rate of malaria, contraction of malaria, went down 50% in many of these communities that used these pyrethroid impregnated bed nets. I'm very familiar with literature, Vincent, because I read it.
Well, you may have read it 20 years ago, but
no,
change.
it wasn't used 20 years ago. So these bed nets are the reason why these insects are now resistant to the pyrethroids because of their widespread use. So I brought in an article from the Chemical and Engineering News, which features another approach.
Read, right? That's the one journal.
This is the one.
That's the one you read during breakfast. I think I remember right.
That's right. I read this every morning.
Why do you get chemical and engineering?
Because I'm fascinated by the topics that they cover.
I think you must have gotten a free subscription years ago.
No. I joined the American Chemical Society because I wanted their environmental and ecological chemical journals, which I subscribed to for many years. And as a result, you have a choice of getting this newsletter as well. So in this newsletter, the nets are still out there, but they don't work anymore because the insects are now becoming resistant, as you can see. So they come up with a clever new approach, something that the insects cannot become resistant to because it's a physical thing. It's not a chemical thing. They've coated these nets with these little beads, which are toxic for insects. And as soon as they touch them, they bring the spores of certain fungi, which are on these little beads, not only to the insect itself, which infects the insect and kills it, but the females don't live very long anyway, but they bring them to the water source where they lay their eggs, and these fungi then infect a certain area. They get into the larvae, and they get into the pupae, and they start to actually affect the numbers of vectors.
So these spores are harmless to the person sleeping in the bed net?
That's correct. They have nothing to do with people
whatsoever.
Sure? They don't infect people. They infect these mosquitoes.
Would you buy this, Daniel?
I'm a little scared of
it.
I'm not sleeping under that.
So that's and they're come on, stop joking. This is a serious issue because if these bed nets don't work, the rate of malaria is going to go right back to what it was before. So they've got the bed nets. Now, how else can we use them to get around this problem of insect resistance to pyrethroids? Because you can't keep changing the formula. You just can't keep doing that. It's too expensive.
So you're saying this study is not necessary because we're not going to use any of these anymore.
Well, they become less and less effective. It's like drug use. You're using drugs on a net as you would use drugs, control program.
Let's let Daniel tell us.
Yeah, absolutely. No, really, the molecular biology here is fascinating.
It's cool. What do they do in this study?
Okay, so as Dickson lays out, this is, well, I was going to say this is an important topic. I'm not sure he laid that up, but I'm going to say this is an important topic because there's this insect treated net, indoor residual spraying, which are part of the pillars of malaria control. But part of the pillars is that they want to understand the mechanisms of resistance, and they want to monitor where resistance is developing.
Right.
And there's a classic mutation that they have identified, and part of the way they go around looking for resistance, they look for this mutation. But these folks say, you know what, we've got to look.
Mutation in the channel gene, right?
Exactly. That makes them resistant to. Well, they say there's several different, I guess, types of resistance. Yes.
Position 360439997, just to be precise, on chromosome 3R. Okay.
Yes. So we are.
It's helpful, Dickson.
And what they go ahead and do is they say, well, we want to see if that's really an accurate test anymore. Right. Does that still predict the distinction between resistant and sensitive? And we also want to see, are there any other resistance mutations developing out there?
Yeah.
So they do really. I thought the study methodology was pretty neat, right?
Yes.
They get to go to Africa. They get to hang out in Kenya, and that's what everyone likes to. Well,
you would like that.
I would like
wouldn't
that.
You?
Yeah, no, that would be good.
And then they go out and they actually collect the mosquito larvae, and now they're going to rear the mosquitoes to adulthood.
Yeah.
And then they're going to check these mosquitoes from these different areas, the larvae, which now are grown into adults, to see, are they sensitive to the poison, we'll call it. And they do this special test, the mosquito distress test, which is really kind of, I don't know. They take these mosquitoes, right, and they observe them, and they look for mosquitoes that are, as they say, knocked down or showing signs of distress.
Right.
And and they say, like, for example, their legs fall off. and which I would imagine
That's
be
pretty stressful.
quite distressing. And then what they're then doing is if their legs fall off, they're going to go ahead and do a gene extraction. It's actually an RNA extraction.
Yeah.
And then they're also going to go ahead and get DNA from the mosquitoes that were not knocked down, did not show distress.
Right.
And then they go through and they do this process of RNA-seq, which, should we explain a little bit?
Sure.
Actually,
Please
Vincent,
do.
you're
So they extract total RNA from these mosquitoes, and in some iterations of this, you can actually get messenger RNA if you'd like, and then you just sequence the whole thing. Now, back in the day when I was being trained, you could never do that. You had to clone the mRNA as DNA and then sequence it, right? What is the face for, Dickson?
No, you're accusing me of being antediluvian, and you're from the same era.
I'm using it for a purpose to illustrate how technology has proceeded.
Indeed.
So we could only sequence individual messenger RNAs. An example was poliovirus. I extracted the RNA, made a DNA copy, and sequenced it. It took me 1 year to do this. And as I told the class yesterday, if you wanted to look at 10,000 different poliovirus isolates, it would take you 10,000 years to do this.
Right.
30 years ago.
Yeah.
Nowadays, we can sequence one poliovirus in 30 seconds.
That's
so
amazing.
10,000 is not a problem. So you can take all the RNAs out of a cell and sequence all of them.
That's amazing.
And what you get is you get short sequences of anywhere from 30 to 40 bases to maybe 100 or so, depending on the technology. And then you bioinformatically, computationally assemble them, and you get the whole expression system of the host, all the mRNAs pieced together. So you can say,
That's fabulous.
and it's quantitative. The more of a specific mRNA, the more reads, sequence reads you will get, the fewer, the less reads. So you can see if an mRNA goes up or down. So you can compare resistant with susceptible mosquitoes and just see how many reads you get for certain genes, and then you can associate those with resistance. It's cool. I've done this here in the lab.
No, I believe you, Vince. I think you're up to date.
And it's very expensive.
And it's very expensive.
That's the thing. Do you guys do RNA-seq in the Goff lab? I think you do.
We do. We've got Yossi, who is who some of our listeners might know, Yossi Sabo. He's got an RNA-seq project about to fire off, and I'm actually in the process of getting one up and going.
When you say expensive, what do you mean by that?
We mean $500 per
one sample.
Per sample.
And usually you run multiple samples in a lane, 8 or something. So it's a few $1,000 you're going to pay. And I don't have that kind of money, but these guys, they're wealthy.
Well, apparently in the Goff lab, we're
Howard
wealthy.
Howard Hughes looks on them, you know. I'm not jealous or anything.
No,
it's not about money.
No, I understand that, but grants usually are.
Yeah, unfortunately.
Yeah.
And so, the price is coming down, but it's, you want to do an experiment and do a lot of time points and replicates.
Well, then, you're right.
You end up cutting corners, and then you can't interpret the data, and then it's worthless. So I had a student you may know who worked on this, and we couldn't afford to do the right array of sequencing, and we can't interpret the data, unfortunately. I can't afford to do it again. No, I don't want you to feel sorry for me. I don't want you to send any money. I'm just telling you, Dickson.
No, I'm going to send mine to the Gorgas.
So in the end, Daniel, what did they find?
They found a couple different things. One of them, they challenged this issue about whether the classically defined resistance gene was really associated anymore or predictive with resistance.
This is the 1014
The
position?
1014.
That's very
the
interesting.
Serine 1014.
Very interesting.
So this is sort of a problem. They're saying, that was whether that was resistant or susceptible. That didn't seem to be telling us the answer. Sort of calling into question the current approach to determining.
This is very interesting. All the resistant mosquitoes had the mutation. They were homozygous for it. And over 90% of the susceptible ones had it too. Boom, out the window.
Arg.
What do they used to say on late night television?
Forget about it. If you act now.
But they did find another mutation, right?
I mean, it is interesting just to sort of pause at that point. We see these maps of resistance across areas of Africa. If it's based upon this, you could say, well, you know what? We're not sure. Well, we're not sure.
Well, yeah.
As we saw here, you can actually have that mutation and still be susceptible to...
Must be something else going on, Dan.
So that's it. So they go ahead and they find some other associations, and then I think they actually find, and this is
Well, in the end, they really narrow it down. We won't belabor it, but they narrow it down to
these sulfotransferase.
I think 5 genes have an expression pattern that correlates with resistance, right? Mm-hmm. And then they had a single gene with a mutation. Was it the...
that was a sulfotransferase was the highest odds ratio?
With the single amino acid change, right? Mm-hmm. Yeah, sulfotransferase, exactly. And
so how does that distribute in their geographic mapping? Did they do that?
Yeah. Oh, as far as then going back and saying where does that map?
They did that. Yeah. The sulfotransferase, right? They catalyze sulfate conjugation of hormones and other compounds in humans and plants, can lead to either enhanced secretion or production of bioactive metabolites. So we don't really know what it's doing, but you can study it. You can try and figure
relate
that out.
Somehow to its ability to be susceptible or not to these pyrethroids, which are neurotoxic.
It does. Yeah. And then there are 5 candidate resistance genes. Mm-hmm. One of them is involved in methylating RNA. Dickson, if you methylate RNA, what would that do?
It keeps it from changing.
It could prevent it from degrading or alter its translatability. So this is a rather broad stroke. Mm-hmm. Lipid metabolism.
It's even broader.
Proteases, a protease, a binding protein, and a cellular.
Whatever cellular protein means.
One of them is annotated as a cellular protein.
Ooh, that's definitive.
Yes. But the cool thing is they had hundreds to begin with that went up or down, and they narrowed it down to 5. Yeah. By actually combining their work with some results from other groups, right?
Mm-hmm. Yes.
Which they narrowed it down. So now, Dickson, you have 6 genes. What would you do if you were running this program, Dickson?
If I had just 6 candidate genes?
Yeah.
Well, I'd make a little probe for each one.
Yeah.
Then I would take those probes back to Africa in a chip, and I would grab pools of mosquitoes from all over the place. But you also have to make one other correlation. You have to make a correlation between finding those mutants and finding an increased level of malaria in the people that live there, because otherwise it's meaningless.
That could be. So you're an epidemiologist, aren't you?
You're damn right.
I would like to know the mechanism of action of these.
Yeah, well, you would, but you could know both of those things then.
So you could, if you had a model system where you could. Yeah. I don't know this field well, but I presume you can produce these channels in bilayers, patch clamping, and then throw the drug on and show its inhibition. So then you could...
Yeah.
But that's just a channel.
Sometimes they use
These are different genes.
They use xenopus for that also.
You could do it in find out a nice system where you could assess the contribution of these to resistance. Not easy.
No.
Because these are very broadly active.
Well, you'd have to narrow it down, wouldn't you? And you can do that in the lab, by the way. These mosquitoes can be raised apart from their natural habitats.
They can be raised?
Sure.
Reared.
Reared is the word.
Can they be schooled?
No, they don't listen very well.
They have such a short attention span. It's like 2 weeks.
That's it.
So here's the key, Dickson. Our results support the idea that many genes may be involved synergistically in insecticide resistance, with their role being modulated by the life history and selection pressure of mosquito populations. Dickson, I have a proposal to deal with this. I think we should just wipe out all the mosquitoes.
Oh, that's very interesting. I think we should put this out to our listeners and ask them to speculate on an evolutionary basis. What would happen if every mosquito in the world were to suddenly disappear?
I could guess one thing. All the species that eat them and depend on them would die. So who eats mosquitoes?
Right.
Bats? Would all the bats die, or would they find something
Vincent,
else?
Now you're thinking like an ecologist. This is good.
Dickson, is that all they eat? But they eat other insects
No,
too,
bats,
right?
they eat many other insects too, but they're great mosquito eaters. And dragonflies eat mosquitoes. They're called mosquito hawks when I was a kid.
All right, so we'd lose dragonflies too. What else would we got?
Well, dragonflies are food for something else. Don't worry about that.
Are you saying
it disrupted the entire food chain?
Turtles all the way down?
I'm afraid so.
You know what that reference is to?
Yeah, I do.
Really?
I do.
I don't know.
Jonathan Swift.
No. Dr. Seuss.
Oh, the turtle, Yertle the turtle.
Yertle the turtle.
Yertle the turtle. Okay, fine.
And you got turtles and turtles, and the guy says, "What's down below?" He says, "It's turtles all the way."
Yes, it's turtles. That's right. Well, in this case, there are many other things. It's all interconnected. So if you wipe out mosquitoes, you're going to upset a lot of other things.
But I would say, right,
you'll also upset the population dynamics of animals that get sick as the result of these diseases, because parasites are population controllers.
Can you give me an example of extinction of a species that has had multi-level effects on other things?
One example?
Well, you can give me two if you want.
Sure. No. Well, a very good one that I can think of right off the top of my head is the extinction almost of codfish and the rise of the lobster industry, because codfish eat baby lobsters. And so at one point there were so many lobsters in Maine you could have given them away. They didn't want them anymore.
Because the codfish was being overfished.
Yeah, their numbers were absolutely almost zero, and now they're making a big comeback.
Why is that?
Because there's a moratorium on catching them.
Codfish?
Yeah,
they're not that good to eat anyway.
They were delicious.
No, cod.
Oh, come on.
I don't know, I had cod last night. It was very good.
But
I was going to point out that the Hawaiian Islands had no mosquitoes, and then Captain Cook arrived and brought some mosquitoes
on
his ship, and they say in the bilge water. Yeah. So I'm sort of pro exterminating, instincting. What do you call it?
Yeah,
when you get rid of. I would like to extinct all the. I am.
What
done
happened
with mosquitoes.
on Hawaiian Islands when that happened? By the way, because I have a follow-up.
Actually, had a huge impact on the bird population.
The mosquitoes themselves, no, but bird malaria, yes. Mm-hmm. So the mosquito came first, which is a Culex mosquito in this case for bird malaria, and then later a Culex mosquito with the malaria arrived, and the next thing you know, they extincted 5 genera of birds. 5 genera of birds disappeared from Hawaii as the result of invasion of Culex and its parasite. In this case, I think it was Plasmodium gallinaceum. I think that was the one. So those are good examples. Yeah. I mean, there's tons of them. Unfortunately, there's tons of examples of them, of things like this happening all the time. It's part of evolution, but you know.
But on the other hand, Dickson, over the eons of evolution of Earth, many species have gone extinct.
Absolutely.
Right? Many.
You bet. Billions.
And we're still here.
We are. But we are causing extinctions.
The lesson is the Earth is resilient.
It truly is, and it may very well be able to get rid of us if we continue to behave this way.
It would like to, because we're really screwing it up.
Yeah, that's true.
Shall we do another case study, Daniel?
Oh, yeah.
Sure.
Come on, bring it on.
Sure.
I mean, you may be running out. I don't know.
No. It's interesting. I have to say, at the 11th hour, I switched cases.
It's okay.
And the reason I switched cases is I was talking to the medical personnel, to the residents, interns, students, and they said, "Oh, TWiP, you must be recording your Halloween special today." And so I don't know if you guys thought about this, but this is our Halloween special. I'm hoping that you can post this on. Will you be able to post it on Halloween?
Yeah, of course.
So when the trick or treaters come, I could have this on like a loop, and they could be listening. Oh. And so I wanted to pick a very Halloweeny case.
There you go.
Yeah.
If you can find one.
And so I went and I said, you know what? I'm not going to present that one I was going to present. I'm going to present a spooky, creepy.
Okay. That sounds great.
Well, our listeners can judge.
Okay.
How well I did. So we have a professor.
Oh, this is your case, Dickson.
He's a professional professor, I will say, in his 50s. And
that's not me.
No.
And he presents with a chief complaint of a lesion on his big toe. He's got a lesion on his big toe. It's the tip of the large toe, and it's nodular. Okay?
Nodular.
He says there isn't any surrounding redness. There really isn't a lot of swelling, but the area is very itchy. It's not described as an ulcer. Okay, I know you sort of like lesions to be ulcers, but this is not.
It's a nodule.
It has a small central black area.
Oh dear.
Which he says is about the size of a pencil tip, and he's concerned because he feels as though it's getting larger. It's been getting larger over the last week. This lesion on his large toe. Yes.
It's getting larger.
Yeah, so he has something growing, and it's spooky.
Interesting. Can we ask questions?
Yes, you may.
Where does this guy travel a lot?
He does travel, actually. Yes, he was at a conference and
where was he?
Spent some time.
Yeah, where was he?
He had been down in Brazil. He had spent a week
interesting.
down there. No, actually,
At
let's
a
see. He'd been there a
conference.
week before he noticed something,
What part
and
of
he'd been
Brazil?
there for a couple weeks.
Yeah.
And he says that he went to this conference, but he also spent some time on the beaches.
On the beaches.
But he reports that he wore flip-flops. So he's telling us. He's telling us.
He didn't say dong.
Yes, he wore flip-flops.
Keep talking.
Okay, what else do you want to know?
I've got my diagnosis, but that's
Okay.
okay.
Were these beaches remote or were they clean beaches?
He actually quite a bit of different variability.
Get to Ipanema?
I do not know. I did not ask him that. Should I have been asking people who go to Brazil whether or not they?
Which part of Brazil? That's what I'd like to know.
It was so there's a big city in Brazil, right?
There's many big cities.
Yeah, Rio is big. Sao Paulo is big.
Yeah.
Bahia.
So I believe Rio was where the conference was. So it was
Could have been to Ipanema then.
okay.
Interesting. Were there dogs on these beaches
He didn't tell me about any dogs on
or
the beach.
animals? I should
Were there
say.
other people on the beaches?
Some beaches there were the crowds. Other beaches not as many people.
And how long after he went to Brazil did he start to notice this lesion?
So he said he'd been back about a week when he first noticed something there. But now, as we hear, it's been continuing to enlarge since
And did
he
you
first
say it was
noticed it.
itchy by any chance?
He says that it was itchy.
Oh,
Very itchy. Actually,
very
he
itchy.
says.
Oh,
Did you examine this nodule?
Yes.
Daniel?
Yes. Any more history before
did
we
it examine you as you examined it? No, this is a good one.
What
This
is
a
his
real good
What is his profession? He's a professor
he
of
is,
what?
He's what we call a triple threat, right? He's a clinician, a teacher, and a researcher.
Like you.
Like
And
me,
an
like me, but older.
much older.
This man I mentioned is in his 50s.
It is. Wow. So that's very interesting. That's fascinating.
How did he eat the local food while he was there? I presume
He did.
So?
He did eat
Did
the
he
local
eat any?
foods.
Did he eat any raw meat? Raw fish? Ceviche?
That's the challenge in Brazil, right? They eat a lot of fairly raw meat, and I think you'd be pretty hard pressed, I would think, to go to Brazil and not end up eating some fairly rare meat.
Yeah, that's true.
Well, I went to Brazil. I've been there twice, and we went to this fabulous restaurant where the meats were in the front and behind glass. They were cooking, and then they would bring you one after another the big slabs and cut them. Right, you would say okay, and they would slice it
It's
into your
fantastic,
plate.
isn't it?
They were all overdone.
Oh, they were all
Overdone.
Oh, that's terrible.
Yeah.
Still tasty, but not raw. That's
Yeah.
for sure.
So,
But
most
after
of
having,
Brazil's beef comes from Argentina, by the way.
is that correct?
It is.
After having done TWiP and TWiV and TWIM for years,
You were
I-
happy that it was.
I actually
overcooked?
don't eat anything anymore.
That's why you're too thin.
I have stopped eating. Whenever I eat something out of my home, I look at it and go, what am I going to get from this? I'll go to let's see, I was just in Ann Arbor and they had some fruits out for reception, fresh cut fruit. I said, no, I'm not going to touch that.
Oh, Vincent.
No way. I'm not going to touch that
You
because
should
the
have brought
skin
your parasitologist with you.
could have been contaminated, and then when they cut it, they introduce it into the fruit.
you're
Forget it.
looking at, I see.
All these things, and so I have a lot of problems eating, but it's okay. Keep my weight down.
Keep your weight.
So he's an adventurous eater. Did he have any sex while he was in Brazil?
He actually went down with his wife.
He did. So that means he had no sex.
No,
But,
Vincent.
Vincent, you're going to have to delete that one.
Why? That's a joke. It's a well-known joke, Dickson. He went with his wife. Okay.
It's a happily,
And his
happily
wife-
married man from
Did his
Vermont.
wife go to the beaches with him?
Yes,
Did she wear flip-flops?
She also had protective footwear,
And did she?
but she's not reporting any
She
problems.
doesn't report any problems.
No, it's just this guy.
Wow.
This guy's unlucky. He
Okay.
encountered some.
Anything else we need to know, Dickson?
Nope, I'm pretty sure.
Any unusual medical history? Is he immunosuppressed?
No, he's actually a healthy guy. He has no prior medical problems. We asked, and he had not had any prior surgeries. He was not allergic to anything. We asked about family history. It was, nothing that we could really pin down.
He's a clinician of what kind of...
I'm going to leave that vague just because, I don't.
He doesn't want to incriminate anyone.
But
I want to incriminate. I want to be like, hey, that's my toe you took.
He's he's not an ID doc because he would have figured this out.
He is not. He is not an ID. He is not an ID doc.
No, this is an oddball.
He's not an ID doc.
Definitely an oddball, and I know why he did this.
And the reason he came is because this was growing and itchy, right?
It was
very
growing
itchy.
and itchy.
Very itchy.
All right, I'm done. Anything
So wait,
else?
I have a joke.
Okay.
Joke.
How do vampires protect themselves from infectious diseases? With immunoglobulins.
Yeah, I've heard that one.
That's a Fozzie Bear. It's a Fozzie Bear joke. No one here even smiled. I want the audience to know that as that joke was being told, everyone,
Yes.
I got this grim face.
I may have heard that before.
Did
some people out there might not.
Daniel, this lesion is confined to the large toe. It's not moving elsewhere.
No, it
right?
is a single lesion,
And a black
enlarging,
spot.
very itchy with a black spot in the center of it on the large toe.
Did you do any blood work? Does he have elevated white blood cells in the blood or anything like that?
No blood work was done.
No, but you made the diagnosis without blood work.
It was done without blood work.
And
Wait,
without,
I do have another.
without
Yep.
biopsies either? No biopsies done?
Is that what you're going to request? This is like you're on, do you want, a call to the audience, a biopsy, a blood test? What would you like, Vincent?
Like a biopsy, yeah.
You want a biopsy?
biopsy.
Well, I don't know anything, so
Total removal.
maybe you could figure it out without a
Okay,
biopsy.
Dickson wants us just to scoop the whole
I
end
want
of his
total
toe off.
removal of this one.
You
So
want to
let me
remove his
ask
toe?
you another. Just cut the toe off and get it over with. No. Describe the lesion a little bit better. There's a black center in it, but there's also another characteristic that I'm looking for here.
Do you want to see a picture? And
Can you? No. If
describe
you
it?
showed me the picture, that would give it away.
That would ruin everything.
You said it was
would ruin
nodular.
everything,
So what is this nodular? About an inch in diameter?
Oh, less than that.
Less than that.
A quarter of it.
And it's on the
It's
anterior
a little
or the posterior?
smaller than. It's about a centimeter in diameter.
Centimeter.
What
He's talking
color
like
is
an
it?
obstetrician. You're 3 centimeters dilated.
Now we're drifting.
The size
about a
is actually
centimeter.
important to get
on
a
the top of
sense
the toe, so behind the toenail.
No, it's really the front of the toe. So think
tip.
of hold your hand out.
the
Put your
tip.
hand on the table, and at the very end,
the anterior portion of the toe.
anterior portion
Was
of
that-
the toe.
And what color was it?
The area. So we have a black center,
do.
and then we have a centimeter. I'll say a donut, which is sort of somewhat blanched, a little paler.
Thank you.
Apparently, that was.
My case rests.
Daniel, what kind of shoes does this guy wear normally? Does he wear open-toed shoes or
No, he wears normal
Closed-toed.
dress
with socks, right?
with socks.
All right, so you did this without a biopsy, huh? Okay.
We did not say that. We're just
Did
you're
we-
requesting a biopsy, and at this point, I
I
have
want
not.
the whole thing.
You
He
want
wants
the
the
whole
whole thing
lesion
out.
removed? No questions asked?
None.
You already feel so confident in what it is that you're just going to okay, wow.
I do.
Okay.
I do.
That's why we keep him in the parasitology lab. I don't know.
So it is swollen, correct?
It's a nodule.
It is a nodule.
So it's elevated from the surface
Yeah,
of
when
the
you
toe.
feel the surface, and you feel, here's this 1
And it's
centimeter
painless.
Very
raised,
itchy.
painless, itchy.
itchy. But when you palpate it, he doesn't scream in pain, right?
No,
It's not
no,
like
he does not scream.
inflamed, right?
No, there's not a lot of redness, not a lot of erythema,
Right.
inflammation, not a lot of pain.
Okay. Very good.
Yes. Do you have any Halloween jokes that you wanted to leave our listeners
come on,
with?
Vincent.
Halloween jokes.
Yeah.
Give us a microbial Halloween joke.
I never have jokes, period. You know that.
Okay.
I'm not, I'm just not a fun guy. What do ghosts,
Fungi?
what do ghosts eat for,
Oh, that was a good one. That was so good. Not a fun jive.
what do you do if 50 zombies surround your house?
What do you do if zombies surround your house?
50.
If 50 zombies surround your
No
house,
idea.
you hope it's Halloween.
Right. You're reading these now.
Why didn't the skeleton want to go to school?
I have no idea.
His heart wasn't in it.
Oh, I like that.
You know that one. That's an old one. Why'd the skeleton cross the road?
I thought it was why couldn't he cross the road? It's because he had no guts.
Yeah, that's right. There's one. Why did the skeleton cross the road? To get to the body shop.
Oof.
Okay.
Why are ghosts so bad at lying?
Gee, I don't know.
Because you can see right through them.
All right.
So there you go. What's
Okay.
the last one? What's a vampire's favorite fruit? A nectarine.
All right.
Not bad.
Clever.
Not
Nick.
bad.
Do you have a joke, Daniel?
No.
Really?
No, I have
Do
not.
any of your children have a joke you'd like to
I
relate
think that
to
if I
us?
told a joke on TWiP and then my kids listened and heard that joke, they would, they'd be
They'd
horrified.
moan.
Yeah, my one was
Horrified,
My one
it's
was
Halloween, come on.
My one was why are ghosts so hungry this time of year?
Oh, because?
Because all the hot dogs are Halloweenies.
You know,
I once was my kids were in Boy Scouts, and we used to go camping, and we would tell around the campfire at night scary stories.
scariest story.
And there was one called Swish Plop. Have you heard that one?
No, Vincent, would you tell
No,
us?
it takes about an hour to tell. It was a lot of fun.
Yeah.
I heard someone tell it once, and then at another camp out, I repeated it. And the thing is, it's all about people sitting outside and glowing eyes in the background, so everyone looks around to see if there's something behind them. Let's read a few emails, okay?
There you go.
Yeah, we have quite a number.
We have a lot here because we haven't done it in a while. Robin writes, camel red blood cells, when mature, do not have a nucleus. We had said, what else besides bird red blood cells?
Mea
don't have
culpa.
that? So
Yeah,
there
well,
you go.
I actually
Yeah.
thought I knew that, but I didn't.
Mea culpa is actually your middle name, Dickson.
Well, it's okay. It's fine.
Like
I'm
all
willing to
mammals,
admit it.
camel red blood cells are without a nucleus. This is a very useful adaptation to increase oxygen carrying potential. However, their red blood cells are a different shape, i.e. oval instead of donut shape. The importance of this is that they can be aligned in the direction of the blood flow, so that a camel is at much lower risk of complications from thickening blood when dehydrated.
That's nice to know. That's a useful piece of information.
Dickson, there's one from Cecilia, which I think you should take.
Cecilia writes, hi, my grandmother and great-aunt were infected with trichinella back in the 40s. The story of them eating undercooked pork is too close to the bone, no pun intended, was repeated endlessly. For the rest of their lives, they were unable to eat pork without getting sick. I have listened to your Trichinella podcast and don't remember hearing you say anything about being sensitive to pork after the infection. I'm wondering if this could have been something they imagined. No, but I think it was psychological. I think, and she also writes, thanks, love the podcast. So I think that anybody who knew that they contracted anything from eating raw or undercooked pork, that point onward, they would think 3 times about ingesting it. So yeah, it's probably a psychological effect of having trichinella.
You think there are such things, Dickson? Psychological effects?
definitely.
Daniel, would you agree?
Yes, I agree. People probably have like an experience like this for like vodka or gin if they ever drank too much in college or something. And now,
I wouldn't have.
It's not. You're not actually allergic, but it just brings back the bad memory.
It truly does. It truly does.
All right. Daniel, can you take Anthony's?
Drug goes from $13.50 a tablet to $750 overnight by Andrew Pollack, September 2015. And there's a link to the New York Times article.
Yeah.
And then daraprin, known generically as pyrimethamine, is used mainly to treat toxoplasmosis, a parasite infection that can cause serious or even life-threatening problems for babies born to women who become infected during pregnancy, and also for people with compromised immune systems like AIDS patients and certain cancer patients. Okay. On a separate and unrelated note, I should have known what a GERD was. But did not. Presumably this is karma from my many years ago deriding a biology teacher for not knowing what a cavy was. Why does Professor Despommier call these animals weird? Is it because of their unusual water metabolism or conservation strategies?
Yes.
Thank you.
The answer is yes to both of those questions. When I say weird, I didn't mean weird. It's a very unusual animal in that it lives in a very dry climate and it doesn't require a lot of water. So there are lots of desert-adapted animals, and this is one of them.
This New York Times article quotes the chief of infectious diseases at Mount Sinai. And here's an example. It's the Icon School of Medicine at Mount Sinai, because he gave money, because it used to be just Mount Sinai School of Medicine.
Yes.
She says, Judith Aberg, do you know her?
Mm-hmm.
What are they doing differently that led to this dramatic increase? She said it will force hospitals to use alternative therapies that may not have the same efficacy. She's right.
Yeah.
Well, they're just trying to make. They probably bought this and they're trying to make back money.
They interviewed the CEO of the company that bought it. His overall goal was to make a lot of money. He was very open about it. Very obnoxious little kid.
He's all about money, like you.
And nothing else. And nothing else. Why did you just use that in the same sentence?
What are you talking
you bite
about?
your tongue there, Dr. Racaniello. Yeah. I'm not all about money. I'm about saving people's lives by participating in TWiP.
Yes, this edutainment.
And I don't get paid for this. Remember that.
No, I was going to say, it's actually, for a while, we've had a problem with pyrimethamine even just getting it. So it wasn't just $13.50 a tablet. It was something we couldn't even get.
He bought all the reserve.
Yeah, I don't think we're going to get it for $7.50 either. And it has forced people to use alternatives. And I have to admit, I've been in situations where we haven't had access, and so we have used other medications, and they're not as well studied. You're not sure the efficacy. It is far less than ideal.
We'd almost pay someone to set up a small chemical factory to make pyrimethamine and get even with this.
I sometimes
gold
think
digger.
That medications should fall under Homeland, under national. No, it's it's an issue, and we do periodically do I should say, like the Cipro stockpiling, the Tamiflu. Certain medications like this are for these individuals life-saving or preventing horrible morbidity when these pregnant women might have an infection.
Yeah. It's the most onerous reason for charging for this drug is just to.
Daniel, if you had a pregnant woman who got infected with toxo in the first trimester, would you treat her with this drug?
No, I don't believe this is first trimester. There's actually, is it spiromycin is what we use? I have to check. But you have to be careful during the different trimesters and use different regimens.
Exactly.
All right. But it's a little dicey either way, treating it first trimester, right? So
we sort of have this rule is depending upon where you are in pregnancy, it affects the risk of transmission and then the severity of the manifestation should it be transmitted. Pregnancy is tough.
Yeah, I think we should eradicate pregnancy, don't you? Then we won't have this problem.
You're becoming a target for a lot of people listening.
We'd have no sick children.
Get rid of mosquitoes and pregnancy, then we have no problem.
There you go.
Oh, but then we eradicate people. Yeah,
no host, no parasites, problem cured.
All right, Steve writes, hi, TWiPers. Interesting to go back through TWiP 95 again to re-listen to the part about arsenious water complicating leishmaniasis treatment. I remember going to an interview for a lab tech position at Bedfordshire, England's Silsoe Agricultural College in the early 80s. The college had a specialty of bringing in overseas students from developing countries to equip them with training and appropriate technology to take back to solve problems in their own countries. One of the problems which was being showcased was the development of easily installed tube wells to solve the problem of villagers having to walk miles for water.
That's right.
These were getting quite a bit of publicity at the time.
Yeah.
I didn't get the job, but it was maybe a decade before the stories began to come in about the arsenic poisoning problem, which apparently nobody thought to check in the enthusiasm to help out with the tube wells.
Indeed.
Now we fast forward to today where we hear the arsenic also makes them difficult to cure of leishmaniasis. The law of unintended consequences writ large. I bet the ancestors of these people had already found out that they couldn't dig wells in these places, but their descendants had forgotten why. Incidentally, on carpet beetles, I used to get sparrows nesting in the eaves of my house years ago. They've largely died out now. There was usually a time each spring when I would wake up feeling something bite my leg and would turn back the duvet to find a carpet beetle. They were coming in from the bird nests and heading straight for my bed. I thought this was a crazy thing for them to do, as they're so fragile that I nearly always squashed them just by turning over. Even if they weren't squashed by me turning over, they were certainly squashed when I found them biting me. And on beluga, I'm quite surprised that you all forgot the 3 beluga that Daniel reported seeing in your own area, which were reported to be diving for bunker. If this wasn't just a one-off, that would be a pretty good place to pick up parasites from cat feces in the East Coast cities runoff. Well, that's true. Remember they found toxo in belugas? But that was up in the North Pole, right?
That's right.
Up around Alaska, right?
That's right.
Always a pleasure to hear of a new TWiP. It's the TWiP podcast that I have most chance of following due to the relatively low level of alphabet and numerical soup of biochemicals, cytokines, genes, antibodies, and lymphocytes that require someone with the ability to instantly memorize telephone directories to be able to keep track. I'm always impressed that anyone can really learn the subject at all these days. They're always good to listen to, even when it does go way over my head. So thanks again for the brilliant service and dedication to educating the masses. Regards, Steve is from Luton, England, where another bright blue Indian summer day is just getting underway, and my grapes might even get ripe this year.
This would be good.
Steve writes, apologies for a deluge of email from my direction today, but I just got your posts from email, and it reminded me of another question I wondered about. In one of your recent podcasts, you featured aphids that were taking up viruses from plant sap, and these were making their way into the aphid salivary gland. What does an aphid need a salivary gland for? It does not chew its food, and there's nothing to lubricate because the plant sap, as I understand it, just pumps straight through the aphid and out the back where it coats the cars parked below with sticky goo, which is then partly consumed by wasps before being colonized with black mold until we got invaded by harlequin ladybirds at last. Has the aphid saliva been modified to dissolve waxy cuticle? I think not, because most go for soft new stems rather than waxy leaves. Do plants get irritated by the punctures and try to plug them and need anticoagulants like other insects do with blood? I've seen this work in reverse with aphids foolish enough to bite into opium poppies. In most cases, though, a salivary gland seems superfluous to an aphid. Just punch a hole and hold on. All the best, Steve.
Actually, there's quite a literature on this that, in fact, it's needed to keep when they stick their, what do you call that thing that they proboscis into the plant, it keeps it from clogging up, sort of like the salivary pharmacopoeia this time of a
there you go
aphid. I looked this up. There are tons of papers, people studying this.
Oh yeah, because they cause great harm to agricultural products, and they want to get rid of them, but trying to get rid of them is very, very difficult.
Here's an article in the Biological Reviews of the Cambridge Philosophical Society called Aphid Saliva.
Ha.
Yeah, all about it. It's full of chemicals.
That's right.
And it activates defensive mechanisms of the plant, keeps the tube open.
Yep.
It's incredible.
It is.
It's so, Steve, it's got functions. What we say is in the nature world, everything has a function, right, Dickson?
This is true. Otherwise, it would be discarded.
Even our appendix, which we used to think had no function. Remember, Daniel?
Yes.
We now know. Who could forget?
That's why I think they call it the appendix, right?
That's right.
that part of the human body that you ignore. You say, I'm done.
All right.
That's right.
We have one more email. Dixon, it is rather long, but I'm trusting.
I'm going to try. I'll give my best. Gene writes, Sirs, I just became aware of your podcast via iTunes. I listened to your first 2 broadcasts this afternoon and am hooked. A question arose during your second hour. I contracted Giardia from a roadside spring in western Washington a number of years ago. Being a mountain climber and mountain rescue volunteer, I had read a good deal about it before getting it and knew that the early symptoms were totally inconsistent. In my case, I suffered bouts of acid reflux, which I had never experienced in my previous life, a general weakness and lethargy. These symptoms lasted for about 2 weeks before the diarrhea hit. The next morning, when the unmistakable symptoms hit, I went to my HMO immediately and got Flagyl to treat it. A few days after I started the drug, I got a call from my doctor. He wanted me in immediately for further blood tests. He said that the ones taken on the day when I went to the hospital had returned pancreatic and liver values, which he felt were critical. I had already begun to improve, though, and I lost about 10 pounds at that time. He required that I have weekly blood tests for the next month to ensure that there were no further damage. I was surprised, therefore, to hear Dr. Despommier say that the total effect of Giardia was intestinal. There is nothing I have ever discovered in the literature to explain the effects it had on me. Generally, the descriptions are limited to diarrhea, weight loss, and related symptoms. At any rate, I'm looking forward to listening to all of the podcasts. I've had my share of parasitic diseases over the years, from malaria to dysentery, having traveled extensively in the third world. Your podcast is certainly one of the most fascinating I have listened to. Great teaching may be gone from the university campuses, but the internet is certainly providing a marvelous opportunity for us lifelong learners. Thank you, Gene, from Bunny Lake, Washington. Gene, that's a great letter, and I will reiterate my contention that, as far as I know from the literature that I'm very familiar with, there are no systemic effects of suffering from giardiasis.
Why don't we ask our infectious disease specialist here?
We now have an on-call doctor who could add to the dilemma by simply confounding it with his own experiences.
All right.
Yeah, no, I'll actually. I'm gonna agree with our emailer that there are systemic symptoms, and so this is the great thing. They've done these studies, and they go through, and they get what symptoms, what have you developed, and I'm going to show this to Dickson as I go through the list. Is that it isn't just restricted to the diarrhea. About 86% of people are actually going to report this generalized malaise, weakness, the flatulence, nausea, vomiting we expect. But people, about 15% will actually develop fevers. And then about 10% will develop this urticaria. And so there seems to be a systemic immune response to this. And Dickson's holding up his finger.
No, I am, because, I don't want to counter argument at all. I want to add to the description, because I think that when you upset the microbiome with a secretory diarrheal disease, as we might see here, but it's actually not secretorial, is it? It's a blockage of fat absorption. So the fats that travel down do not get into your body, and therefore there should be a systemic effect of the loss of absorption of normal lipids, because they're essential for life itself. But what happens when they get down to the large intestine? They start to alter the microbiome, and the microbiome changes dramatically, and that's where the flatulence comes from. Now, tell me that weight loss doesn't have an effect on your sense of well-being, and it makes you lazy.
He's turning it around. He's the one who's no systemic effect. Now he's explaining.
I want to know why you think malaise is a systemic symptom when you're actually malnourished as a result of not absorbing your fats.
It's not just malaise, other things.
Yeah,
as
but
well.
that's not a sign that's...
Anyway.
I mean, I'm not a clinician, but I would argue that there's no tissue invasion of this parasite.
Yeah,
so
it doesn't have to. Cytokines can be produced that.
No, these are secondary effects. These are secondary effects. I would agree there. I would agree.
Well, I don't know if secondary. Yeah. It doesn't matter. A lot of diseases this happens.
Well, what happens when you deprive someone of their essential fats?
Yeah.
And that's what this organism does. And so that's the systemic effect of this.
Do you think great teaching is gone from university campuses?
No, I don't think so at all. Of course not. I think there are many passionate people who love to teach, including somebody sitting opposite me in a pink shirt who is recognized the world over as a great educator. And I can tell you this because about 3 weeks ago, Dr. Vincent Racaniello was asked to join the Leipzig online teaching corps, and you recorded a whole bunch of sessions for them, and we anxiously look forward to them.
So, Dickson, this is why these podcasts are great. We reach people, we can teach them. And in the beginning, you didn't believe any of this.
Well,
yesterday I gave a lecture to a course on emerging infectious diseases.
I used to co-direct that.
These are mostly students in the School of Public Health.
Mostly, yeah.
And two of them came up to me afterwards, and they said, "We've been listening to TWiP for 5 years, and it's why we came here.
Excellent.
We heard of Columbia, and we thought it must be a good place."
Cool.
Isn't that nice?
They came because of
because they heard of TWiP, and they said, "It's at Columbia. That must be a cool place." So they applied, and they liked it, of course.
Good.
But they have nothing to do with me. But I give this place an aura. And we give it an aura by doing these podcasts. Do you understand?
I do understand.
This information emanates from here. People love it. And now, do you think that any administrator here at Columbia appreciates this? No.
They don't even know what a TWiP is.
In fact, outside of our little circle, no one cares here.
No, that's not, come on, I wouldn't go that far.
Who cares? Do you think our dean cares? No.
That's a leading question.
My theory is they think it's so great, so excellent, that they don't want to mess with it. They're afraid that if they intervene...
Exactly. That we may actually ask them to pay us for doing this.
Well, somehow our passion might somehow be diminished or corrupted.
That's okay. I love
That's what it is.
I love to complain. It's all right. We're fine. We are doing just fine. This episode of TWiP, like all the others, will be found at iTunes and also at microbeworld.org/TWiP. And did you hear, probably you guys don't know this, the other day, 2 days ago, Google announced that they are now going to be carrying podcasts on their online music place, Google Play.
Okay.
And that's good because we can get more listeners.
You betcha.
Because right now, iTunes, Apple has been the biggest repository of podcasts,
Right.
for which we have been very grateful. But now Google has seen. But you know why they did this? Podcasting is becoming very hot.
Is it now?
Yeah, the big names are getting into podcasting.
Or are they?
It's moving away from the amateur hour, which people call us.
So what's a big name in your opinion? Who's gotten into this recently that you think everybody should know about?
No, I don't want them to know about it. I want them to focus on us. All the big radio stations make podcasts. They have a lot of money to throw behind this.
Yes, they do.
They have studios. They have producers. They have people who help.
Quality equipment.
I have to say, don't you think it's quite a coincidence that we're about to record TWiP 100 and Google is putting podcasts on? I think it's a coincidence.
Anyway,
we've been discovered.
We don't know when they're going to release this, but I yesterday registered all of the Twix podcasts on Google, and they have to be accepted, of course. But they should accept us because we're a quality product. As
we have a long track record
from our listeners.
That's right. Long track record.
And it's very cool. We have all these great teams of podcasters here, and it's wonderful. And if you have questions or comments, we love answering them, especially if you correct Dickson. We like that a lot.
Vincent, you're going to get lots of email about just stop picking on Dickson, would you please?
TWiP at twiv.tv. Please, TWiP, TWiP. Sometimes some of you send them to twiv at twiv.tv. I know it's confusing, and I'm going to change that soon, by the way. We're going to fix it. But if it goes to TWiv, I don't see it. And that's why sometimes we miss your email. Okay? TWiP at twiv.tv. You have that, Dickson?
I do.
I don't have to ask Daniel because I know he gets it. But Dickson, TWiP at
twiv.tv. Say that fast 3 times.
Daniel Griffin is at Columbia University Medical Center.
And I guess
Daniel.
Presbyterian Hospital. Just
yeah,
to keep
right.
it all
That's
straight.
right.
They haven't changed the name recently.
Thank you, Daniel.
Oh, it's been a pleasure. Thank you.
Maybe someday to be the Griffin Columbia Medical Center. Who knows? Dickson Despommier is at tricanella.org, medicalecology.com, verticalfarm.com. Thank you, Dickson.
You're welcome.
The father of vertical farming, the potter of vertical farming.
And so who was the mother?
Parthenogenetic.
Parthenogenetic of a father?
You see what we say when there's a father there.
Well, I was told that by somebody. How can you be the father of something unless there's a mother involved?
Well, it is a metaphor, right?
Metaphor. It's a metaphor.
You gave birth to something, but it ignores the fact that fathers don't give birth to anything, right?
It's correct.
Anyway, thank you very much, Dickson. I appreciate it.
It's been a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiP is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP
is parasitic.